Hi, my name is Michael Hilliker, and this is my reproductive system portfolio. So I did my presentation on the determination of sex and the role of genes versus hormones in that determination. So I felt that it was important to start off with a little bit of background information just about how everything develops. So the human genome programs sexual development. So humans have 23 pairs of chromosomes, or 46 total chromosomes, that will guide this development. So the genetic sex of an individual will depend on these sex chromosomes. Females are XX and males are XY. Now, in the absence of a Y chromosome, an embryo will develop into a female. So the male and female sex organs are similar before they differentiate. So they consist of gonads, internal genitalia, and external genitalia, just in a broad sense. So internal genitalia, they include all the accessory glands and ducts that will connect the gonads with the outside environment. And the external genitalia is everything that is an external reproductive structure that is visible. Now, males and females both have gonads, as I mentioned. The males have testes, and the females have ovaries once they differentiate. So now getting into some of the actual genes and hormones that will determine gender and development. So the determination of sex between male and female depends on the presence or absence of the sex-determining region of the Y chromosome, or the SRY gene for short. So I have a little picture here off to the side. You can see just that it's pointing at this region. So if the SRY gene over here is present and functional, the bipotential gonads will develop into testes. So we'll guide it into male development. Now, if this SRY gene is absent, the gonads will develop into ovaries under the direction of multiple female-specific genes. Now, moving towards more of the male embryonic development, so this would be after that SRY gene is present. So the SRY gene will produce a protein that is called the testes determining factor, or TDF. So this will bind to DNA and activate a few additional genes. Some I have listed are SOZ sub 9, WT sub 1, and SF sub 1. Now, the protein products of these genes and other genes will direct the development of the gonadal medulla into an eventual testes. So it's also important to note that testicular development does not require male sex hormones, such as testosterone. The developing embryo is actually unable to secrete testosterone until after the gonads differentiate into testes. Now, moving on from there, there are 3 hormones that are secreted from the testes once they differentiate. So they are the Sertoli cells, and these secrete a hormone, well, a glycoprotein, anti-Müllerian hormone, and there are also testicular interstitial cells, or Leydig cells for short. They secrete androgens, testosterone, and the derivative of testosterone being dihydrotestosterone, or DHT for short. Now, some important points with the female embryonic development. So this is, again, at the point where the SRY gene is absent. So once that occurs, the cortex of the bipotential gonad will develop into ovarian tissue. And there are multiple genes that are required for this development for the functional ovaries. So I just didn't have them listed, but there are a number of them. Now, in the absence of testicular anti-Müllerian hormone, the Müllerian ducts will develop into the upper portion of the vagina, the uterus, and the fallopian tubes. And also, without testosterone being present, the Wolffian ducts will degenerate, so they won't be there anymore. And also, without dihydrotestosterone, or DHT, the external genitalia will begin to take on female characteristics. So these last two slides are more of a summary of when and where all of these genes and hormones will interact, whether they're present or absent, and cause the actual development of male to female structures. So if it's a female, at 6 weeks, the gonadal cortex will form the ovary, the gonadal medulla will regress, Wolffian duct will regress because the testosterone is absent, it isn't there, and the Mullerian duct will become the fallopian tube, uterus, cervix, and the upper portion of the vagina, and the anti-Mullerian hormone is absent. Now, if it's a male at 6 weeks, the gonadal cortex will regress, the gonadal medulla will form a testes, Wolffian duct will be present, and it will form the epididymis, vas deferens, and the seminal vesicle, with testosterone also being present. The Mullerian duct will regress because of anti-Mullerian hormone being present as well. So more of all of the internal structures are developing in the male and the female at this point. Now back to if it was a female, at the 10-week mark, the gonadal cortex will become an ovary in the absence of that SRY protein, and under the influence of those multiple female-specific genes, the absence of testosterone will also cause the Wolffian duct to degenerate. Same thing at 10 weeks for a male, some of the development. The SRY protein in a male embryo directs the medulla of the bipotential gonad to develop into testes, and the anti-Mullerian hormone from the testes causes the Mullerian ducts to disappear. Now, once birth is close to occurring, if it's a female, at birth, the absence of anti-Mullerian hormone allows the Mullerian duct to become the fallopian tube, again, the uterus and the upper part of the vagina, so more of a summary of what's occurring at this point as well. If it's a male, again, the testosterone will form the testes, well, the testosterone from the testes will convert the Wolffian duct into the seminal vesicle, vas deferens, and epididymis, and dihydrotestosterone controls overall prostate development. Dihydrotestosterone also causes the development of the male external genitalia, and in the absence of androgens, the testosterone and dihydrotestosterone, female external genitalia is produced. Now, the diagram over here on the right, again, is a nice summary picture of the gonads and actually seeing the Wolffian duct, Mullerian duct, how they are present, how they end up forming into the females, the ovary, the oviduct, uterus, and the cervix, and without male hormones, and then looking over at the male side with the male hormones being present, seeing that the epididymis, testes, vas deferens, and seminal vesicle, where they all are and where they develop from, is a nice representation of seeing all that. So I also have my worksheet here, just to take a look at this as more of a study guide. So I did it with matching terms and just saying that terms may be used once, twice, or not at all. So a couple of the questions that I have, we'll just run through all of them, actually. So the number of chromosomes in the human genome, I put as being, the answer is 46. So I put 46 and 23 there. Which gene would turn the bipotential gonads into testes? Would be the SRY gene. The Sertoli cells secrete anti-Mullerian hormone. Leydig cells secrete testosterone and dihydrotestosterone. And in the absence of DHT, will cause the external genitalia to take on female characteristics. Testosterone, the absence of that will cause Wolffian ducts to degenerate. And AMH, being absent, will cause Mullerian ducts to develop into the upper portion of the vagina, the uterus, and the fallopian tubes. DHT will promote the development of prostate gland and external genitalia, and the SRY gene, being absent, will cause the gonads to develop into the ovaries. And that will wrap up the consideration of genes versus hormones, and their role in the reproductive system and its development.